It's a pleasure to introduce the last speaker for today and for the conference. Dr. Achintya Das is the Deputy Director of Research Informatics at Syngene. He's over 2 decades of experience in computational chemistry and biology, multi-scale modeling and simulations, discovery informatics, and AI applications in pharma, consumer goods, and other domains. Prior to Syngene, he was Director of Life Sciences and Computational Chemistry at Strand Life Sciences, and he set up the Supercomputing Center for Bioinformatics and Computational Biology at IIT Delhi. Today, the presentation is entitled Enabling Data-Driven Drug Discovery from Target Identification to Lead Candidate, and we'll listen to that presentation now.
Hi, I'm Achintya Das, responsible for Research Informatics at Syngene. And today I'll be talking about how we use data to drive drug discovery, starting from identifying the right target to a lead candidate. At Syngene, we look at informatics as a way to integrate diverse computational techniques across computational biology and chemistry, informatics, AI, to drive these decisions that you typically take along a drug discovery program. We integrate data that we generate on a project and learn from past data to enable those key decisions that you take to find which is the right target for your therapeutic area, what is the right molecule for that target, and how does the data enable you to increase success rates in clinic. At the beginning of your program, we look at integrating omics data with various information that's available around the therapeutic area, combine that with network biology to establish a connection between the disease and the target, come up with mechanistic hypothesis, look at various druggability parameters to ensure that the targets that you choose have the best chance of success. Once a target is identified, we are now looking at what is the best molecule for that particular target. The best molecule needs a fine balance of properties that makes it an ideal lead candidate, which would mean it has to have the right activity, it has to have the right PK parameters, and it should be safe. The multi-parametric design workflow that we have put together enables you to do that in silico before you test out the likely candidates. Moving on to preclinical development, the same models that we build to identify targets now can be used to assess the safety profile, look at biomarkers and signatures for that particular program, and that enables you to identify the right molecule to progress. And once you move to the clinic, the same data and the same model is used to stratify the patients, perform responder, non-responder analysis, adapt your trial for the best chance of success. Target identification starts with bioinformatics. What we do is use NLP to mine all the data or all the information that's available around the therapeutic area of interest. We then choose the right data sets to analyze. And this could be a combination of data that is available in the public domain, or it could be data that is generated by you. And now the data is combined with all the other information to create a knowledge base around the therapeutic area. And this enables you to form a disease network, perform various kinds of network analysis on that to draw connections between various entities and the disease of interest. Overlay experimental data to see how is the experimental data explained on this network. And that allows you to set the context for the experimental data, generate the various hypotheses that would link these potential targets to disease. That again sets the tone to overlay that data over a protein-protein interaction network, where we have incorporated the PPIs, the diseases or the pathways associated with these proteins, drugs which are known to be associated with these proteins, and analyzing that on this network allows you to find links with disease, to find other links which may lead to adverse events, to find what are existing knowledge around a certain neighborhood, and this allows you to find a robust mechanistic rationale about why a particular protein would be of interest as a good target for the therapeutic area. Now, all proteins that modulate your disease need not be good targets. They might have liabilities. The expression profile may prevent it from being an effective target. So, to identify and prioritize targets, you need a holistic view of each of those potential targets. So once we have a list of candidate targets from the mechanistic study, we then assemble a lot of information around those targets. Look at where they sit on the networks. Look at the expression profile available in various databases. Look at what are their homologs, what are the paralogs. And this enables you to get a understanding about where does that particular protein lie in the network. What is whether that family has been druggable. Are there other concerns that you might have, which will prevent it from being a good target? That could be on the network. That it modulates the other pathways. It could be that it shares homology with a lot of other proteins that can lead to selectivity issues, and a combination of all that is used to score the targets based on their strengths, weaknesses, and opportunities. So we use a multi-parameter scoring algorithm that takes into account all the parameters that we discussed and creates a score of the target, and this enables you to then pick out the top targets for validation. Because the entire study is mechanistic, you can use the mechanistic hypothesis that we extract to come up with a screening tree that can help you validate the targets. Once you have prioritized the target, we now need to decide about what is the chemical series that you need to consider to find the right molecule for it. We have created a workflow that integrates data with chemoinformatics, computational chemistry, to identify the right hit, prioritize that based on novelty, synthesizability, ADME, and safety, to give you a focus library as the starting point for your chemistry. So the method allows you to combine various approaches, scan large amount of chemical spaces quickly, because it is not dependent on structure alone. You can actually learn from phenotypic screens and expand the hits. And at the end of it, the focus library is balanced not only for activity but for safety and ADME, so that you have a higher probability of succeeding. So we use machine learning, structural methods, as well as system biology to combine various properties to predict ADME and toxicity parameters that we use in our workflow. For example, for stability, we have models that are based on fingerprints. Properties like cardiotoxicity are extracted from binding models combined with machine learning. For parameters like skin sensitization, which are dependent on electronic properties, we have models that are based on reactivity parameters. And for hepatotoxicity, we have a systems model, which is a mechanistic model that takes into account the modulation of various proteins in the network to arrive at a quantitative and mechanistic prediction of hepatotoxicity. So depending on where we are in the discovery program, the right combination of these models is used to tag liabilities and take decisions. Once we have identified the series and we start generating data on a project, we use project data to guide lead optimization. The data generated in a project is fed into the workflow that builds models that enable you to predict the various parameters that you're optimizing on the project. The workflow takes in a project objective and then does virtual enumeration to look at to score each molecule against the project objective. Look at how close it is to fulfill that objective, and then makes replacements and runs through a virtual optimization cycle to come up with optimized molecules which have the best balance of the properties that you need on that project, which you could then take up for assessment. I will illustrate that with a case study. The problem that I've shown here is optimizing VLA4 antagonists, and these in this project, the several nanomolar compounds were identified for VLA4, but most of them suffered from poor clearance and bioavailability. And the best compound that they could get to get around the poor PK had stability issues. So the question was whether we can use informatics workflow to come up with a novel design that can get around these problems. So in this case, because it was a lead optimization problem, around 200 molecules had already been synthesized on the project. We took the data of those 200 molecules and put that through our pipeline. So the workflow learned from the project data to predict each of those properties. The project function then used the models to score the molecule against the ideal profile that was defined. And what we found was that 75% of the compounds that were made on that project scored less than 50% for the project objective, which means that 75% of the molecules that were made on that project were actually very poor for achieving the project objective. So most of them had very good activity, but these were they had problems with clearance and solubility and other factors. And only 3 molecules had a score of more than 70. So learning from that, we set up a optimization workflow where on that series, R group database was used to enumerate molecules. The models that were built on the project data was used to predict various properties. Those predictions were fed into the scoring function, which gave a score for the molecule, about how good that molecule is for that particular project. And this cycle was run till we could not optimize the score any further. So on the right, you see a table that shows how that score progressed, and the highest that we could get to was around 82%. And the top molecules from there were chosen for synthesis and assessment. And we found indeed that among those top scored molecules, we could get to a molecule which had good activity and clearance rates with high stability, and that met the project objective. So in summary, the data-driven process that I showed here integrates and learns from genomic, proteomic, and phenomic data to enable smart decision making in drug discovery, learning from the project data and combining that with historical data to help you take decisions at every step along that process. It helps identify the right target by understanding biological systems and diseases, looking at mechanism action, integrating that with intelligence around targets to ensure that you identify a target with the best chance of success. It helps you find the right molecule by probing the right chemical spaces, looking at the safety and PK profile, and designing molecules which have the best balance of the properties that you require for your project. And the same models that help you do this can be used for translational research to improve success rates in the clinic, to predict in vivo outcomes, patient response, and help tune your clinical trial for success. Thank you.
Thank you again, Dr. Dus, for your presentation. And if your audio is now working, just let me know. I can see you are muted.
Yeah, it is.
Awesome, wonderful. Good to have you. It was a great presentation. And thank you again for the overview in terms of your drug discovery capabilities, Dr. Dus.
So I think I'd like to ask a question. I mean, great presentation, Dr. Das. I thoroughly enjoyed it. Have worked with Syngene in the past at a previous company, so I'm aware of the capabilities of your organization. With regards to this, I guess my question is really, is there a point at which there are too many properties of the molecule that you want to improve in a program such that even your amazing platform would not be able to actually overcome all of them?
So it's not about the number of properties. It's more to do about how much data you have to do a good job of predicting those properties. Outside of pharma space, we've applied this method to problems where we've had to balance 10 properties. So it depends on the kind of data that you have generated and how well you can predict those properties while evaluating new designs.
So if you have a limited data set, it's much harder to get an overall positive outcome. Is that what you would say?
Yes. So there are two aspects to looking at multi-parameter optimization. So one is when you start a project and you're looking at various molecules across the chemical space. At that time, your focus is to have a model that can do a good job across a wide gamut of space. when you come towards lead optimization, now you're focused on a particular series, and your models have to be able to discern those small changes that you make to be able to predict what that effect would be. So the models that you use at the beginning of a project and toward lead optimization, they're different.
Mm-hmm.
And sometimes what happens is, if you don't have enough data during optimization, you might end up getting very poor results because your model just can't make out the differences between those molecules in the same series.
Right. Thank you very much. Sam, are there any other questions?
Let me just double check here. Nope. And again, if you're interested in connecting with Dr. Dus and his team, you can definitely reach out to me or use our meeting scheduler grid to connect with, I believe, Davina, their company representative. And I do want to take a moment now as we conclude our program to thank our chair for today, Dr. Karen, for her amazing effort in staying the course with us, all the back-to-back presentations. I'm not sure if she even had lunch today, but we are very appreciative of her remarks throughout the day. I want to thank all of our speakers, our attendees, and all of our panelists from yesterday for participating. Again, the whole purpose of this conference, designed by my team, Sherry Monroe, Victor Sheriff, and Luke Castro, was to bring all of us together. All of the talks, hit us all differently, gave us all different ideas. At the end of the day, our motto is precision for collaboration is eventually precision for medicine. Without collaboration, there is no medicine. So I will be concluding this summit. Feel free to network amongst yourselves, message each other in the lobby, in the booth area, in the lounge area, etc. Visit our website for upcoming events as well. I know we have an East Coast-based event coming up late May, early June, so stay the course for that one too. And again, with that said, thank you all for joining me the 2 days, and thank you again, Dr. Karen, for your amazing help and support.
Thank you for inviting me and allowing me to chair today. It was really enjoyable, and we had a lot of amazing presentations.
Thank you so much, and feel free to follow our page on social media. Thank you again.